Hello. Welcome back to What is a Biology? I'm your host, Mr. Johnny Hopkins. Let's dive right on into some DNA replication and repair. So, we all know that in cells, you have DNA, and that DNA has to be reproduced every time you make a new cell, so that cell can do its job. Alright, but how does it do it? So it does during mitosis, but there's this whole question a long time ago of whether DNA replication is conservative or semi-conservative. So when you have your DNA, you have the two strands right next to each other. When you go to the progeny, to the daughter cell, you can either have, is the question, do you have the distribution be two daughter strands and two original parental strands in the two daughters, or do you have one parental and one daughter strand in one cell and one parental and one daughter strand in the other cell? That was a whole huge question a long time ago, and they found it out. It's a semi-conservative model, such that when you have a single DNA, the duplex of the two strands, you get two strands with one of them having a new guy attached to it and another one having a new guy, a daughter strand attached to it. So the new one that is made is called the daughter strand, and these guys are made by something called DNA polymerase. And DNA polymerase goes from 5' to the 3' end and uses DNTP, so like ATP, GTP, CTP, and TTP. And there are a lot of different types of polymerases. Typically, there's the three that we looked at: alpha, delta, and epsilon. Each of these are used in their own specific ways. They have different error rates, and that can be useful in different situations as well, such that you can get a robust, quick strand if you need it, or you can and then have it followed by other polymerases to get it all going. So, replication, it's not done just purely by the DNA. So what actually happens here is RNA is necessary. RNA primers are made by RNA primases, which are a special type of RNA polymerases, as we discussed previously. So we have this weird RNA polymerase have to attach to the DNA, separate the strands, make the bubble, and then make a few nucleotides, and then the DNA polymerase comes on in and attaches to that primer and continues chugging along. So DNA polymerase doesn't do anything by itself. It requires RNA primase first. So when you have a primer, then the DNA polymerase will just keep going from the 5' to 3', making the cognate Watson-Crick strand that mimics the previous strand. And then what happens on the other guy? So as we go from 5' to 3', but then if we look backwards, we have to go the opposite direction. But when you have replication fork, so essentially where the replication ends, you have essentially a free strand then hanging on, doing whatever it wants. And that's not good in the cell, because that means that you can have a lot of nucleophilic attack, and just bad things can happen, man. That's not good. So because this is dangerous situation, what happens is you have DNA polymerase go backwards on it. As this guy's going forward, it's always exposing more and more free nucleotides that are unbound. So you have a bunch of DNA polymerases going backwards and backwards from the 5' to the 3' end in order to have this happen. And so the leading strand is the guy that just keeps going without any problem, making the complementary strand, whereas the lagging strand is the one that goes backwards. So you have an RNA primer always just kind of get made, and then DNA polymerase continues on until it hits the previous strand. So these little guys right here that are attached are Okazaki fragments. So these are discontinuous fragments of the lagging strand. And then these guys are actually connected by ligases that connect all these Okazaki fragments into into a good wholehearted chain that complements the previous one. So you have now two daughter strands and two parental strands all connected with one parent with one daughter always, with a non-conservative mechanism. So one thing to note here is that because you have this primer, you have a few nucleotides at the end of every single DNA replication that you're always going to lose a little bit of information every single time you replicate because you don't have that. So there are a lot of different techniques that DNA just has a bunch of extra nucleotides at the end, so this way you don't lose a lot of information and it's not going to actually come in as a factor really in your lifetime, because when they do experiments on rats, they find that it takes around 3 to 4 generations for actual effects to happen in their lifespan. So now going forward, that's how DNA is made. But DNA also has something weird. You can hurt it, and it gets hurt a lot. One of the major things that hurts DNA is ultraviolet light. So when you go to the beach, kind of hurts you. Kind of hurts your skin when you get the sunburned and you look redder than a lobster at my Uncle Jimmy's barbecue. So ultraviolet light damages it and breaks it. And so there's a lot of damage that just happens every single day, independent of it. Even if you're really in ultraviolet light, you can just constantly, your DNA is getting damaged. So there are a lot of different ways that DNA gets repaired. One thing to note is that when you have a mismatch, there's a specific pathway for that. And one of the common mismatch is a GT mismatch. And that's because usually the thymine is added because since G can do an okay job of connecting with T, so usually it'll just change the thymine to cytosine, and all is well in the world by this protein that just slides along and finds it. But then you have to, what happens if I just say break it? I just break it in half. Then you have to actually do some real hardcore repair. And this repair can be non-homologous or homologous. Non-homologous means essentially you have a DNA strand that gets broken, and you have a different DNA strand, and then you just kind of connect the two. That's not good. That's not good. Because what happens is you're connecting two DNA strands that have nothing in common with each other. And so what happens if you've got a protein start here, and then it just kind of keeps going on and on and on, and you make this mRNA, and then you get this weird protein, and you find out it actually hurts the cell and makes you cause cancer. That's not good. That's not good. So instead, typically the favorite mechanism is homologous recombination. So the major thought behind this is when you have DNA in the eukaryotic cell, in our cells, us humans, we see DNA be slow. You've got this very cluttered environment inside the cell, and inside the nucleus it's even more cluttered. So these guys can't really move on their own. They want to be wiggly boys, but they can't. They're connected to way too much. So the broken DNA stays nearby, so you kind of can sense this very quickly, where the end of one broken strand is with the end of the other. So then recombination is this exchange of strands on separate DNA, so you can have recombinant DNA that connects back with itself, so you have good DNA and you can stay alive and not have cancer. So one thing that's also noted is that this thing happens a lot in mitosis, where you have crossing over. So if you take, because you have two parents, so you have 2 chromosomes, and then so they just kind of will switch over with each other a little bit at some points, and that will cause some genetic diversity to make you a little more different from just being purely the same thing as your mother and father combined. So, what are these things that are going on? So, in UV light, your DNA breaks. So, what happens is you have a strand that is further out than the other typically. And so this leading strand will get attacked by this guy Rec A or Rad51. And these guys are a family of proteins that find broken DNA strands and attach to them. And then what happens is they find a homologous DNA molecule. And they'll invade that strand so that you can have this hybridization so that they're actually connected with each other. And then they can go on and reproduce themselves into recombinant DNA so you don't have cancer and die. So what happens is you have some strand invasion right here. So this guy invades this guy, and so you form a holiday structure. So that's just a crossing over of two nucleotides. It's not like it's just this single crossover of the backbones. And so then what happens is, so this guy will form base pairs with this structure, and this guy will form base pairs with this structure. But there are two different types of mechanisms that go on here now. You have DSPR, double strand break repair, which is very common in mitotic cells. So typically, you're really just going to study this system, this DSPR, versus there's also SDSA, synthesis-dependent strand annealing, which is typical in meiosis. And so what happens is essentially you just form this, reform this guy with some, you reform the proper Watson-Crick strand, you regain the original sequence, then you have it joined back with the original strands. It goes and it ligates and makes all that new DNA. So you have the original DNA as well as the parental DNA just kind of being by themselves as they once were, and everything's good and well. But typically in cells, this is the guy that happens. Double strand break repair. So you have all of these guys finished by just forming the Watson-Crick base pairs. So you have gained the original sequence as found because you have this guy crossing over and this guy forming the Watson-Crick base pairs with that, and that guy with that. So then what happens is you have crossing over occur usually, such that you have parts of one of the parental and parts of the daughter, the homologous structures actually switch over such that like you no longer have the original DNA strands attached to the same original DNA strands. It's this really weird fancy thing that you no longer have the originals. You have a weird ship of Theseus sort of scenario here with that this DNA is made into a new one because it's combined by old ones together. So that's in general homologous recombination and DNA replication repair. So real quick summary. When you reproduce DNA, you go from the 5' to the 3' end, and DNA polymerase makes the leading strand. But then you also have to go backwards and make Okazaki fragments that are caused by these RNA primers and ligated by some ligases. Then DNA is very, very finicky, so it can be extremely damaged, but it's less finicky than RNA. So you have homologous recombination where you invade nearby homologous RNA strands in order to regain the original sequence, and you can have crossing over of these DNA molecules that by this formation of recombination. So that's all for today. Thank you very much.